please, it'll be an honor to introduce you, to our audience. So Dr. Huma Amar is the Senior Vice President and Head of R&D at FSD Pharma. She's worked in the clinical research field for a number of renowned institutions, including Yale, Harvard, UPenn, served in key roles in affiliated hospitals like Thomas Jefferson, University CHOP, Children's Hospital of Philadelphia, Hospital of the University of Philadelphia, and so on. You've seemed to have had a lot of leadership positions, throughout your career: Director of Physician Research Programs, National Public Health Physician, National MSL, Senior Research Business Manager, and lead postdoc fellow in a number of national clinical trials. It looks like your background is actually not limited to any therapeutic area as well. It's been across the board in oncology, rheumatology, dermatology, neurology, cardiology, hepatology. You've named it, you've touched it. Your topic today is on FSD Pharma's 201-03 COVID trial. Very apt and timely to discuss the challenges and success. So without further ado, Dr. Amar.
Good morning, everyone. And Dr. Lakan, that's incredible. Thank you for such a kind introduction. I would like to say good morning and greetings, all the viewers, presenters, panelists and attendees. As Dr. Lakan has mentioned, I'm Dr. Huma Qamar. I am the Senior Vice President and Head of the R&D Program at FSD Pharma. I'm quite thrilled and excited to be here today. This seems like a cool platform, and I think Precision Global has done a fantastic job in gathering all the outstanding leaders on one platform. So I would be sharing some experiences from our novel trial. As you can see it on the screen, it's a randomized, double-blinded, placebo-controlled, multi-center study. It's a phase 2A study where we are utilizing FSD 201 compound with standard of care versus standard of care in the treatment of hospitalized patients with COVID-19. So let's dive deep into what it is. And actually, hopefully you guys can see my screen. I have my contact information here. So in case if you don't get a chance to connect now, you feel free to reach out to me with any questions. So we will be talking today about the FSD 201-03 study objectives design. I will be briefly touching upon the primary and secondary endpoints and inclusion-exclusion criteria. Also, we'll be talking about the mechanism of action and safety profile of our magic compound, which is ultra-micro PEA. And I will go a little bit in detail. I will also talk about the recruitment enrollment challenges and effective strategies. What is the ever-changing standard of care landscape looking like in the COVID-19 trials? And last but not the least, what are the lessons learned? So as you can see it from the title, it's a phase 2A study, and it's a randomized, double-blinded, placebo-controlled study. We are giving it with ultramicronized palmitoyl ethanolamide, pretty much with standard of care versus placebo, and standard of care in hospitalized patients with COVID-19 that fall under the criteria of moderate to severe. The participating sites there are almost approximately 50 sites across USA, Latin America, and Bulgaria. The target enrollment is it's pretty exciting goal of 350 patients that are going to be randomized with, and they are being randomized currently in 2:2:1 format. There are 3 arms. The first arm has 600 mg of FSD 201 compound. We'll be giving twice daily with standard of care. And Arm B has 1,200 mg that we are giving 2 into 600 mg tablets twice daily with standard of care. And Arm C is pretty much placebo with standard of care. So what are the study objectives and what we wanted to achieve? We wanted to measure the effect of ultramicronized PEA, which is palmitoyl ethanolamide, with standard of care, and especially versus placebo and standard of care on day 28, which is considered as the day for disease progression in the confirmed cases of coronavirus, which fall under the moderate to severe criteria, which I will touch base a little later. Also, we want to measure the effects of FSD 201 with standard of care versus placebo with standard of care on disease resolution. So additional secondary objectives were to look at that day 28 mortality rate and other COVID-19 related characteristics, which I will also discuss in detail in a little bit. We wanted to look at the safety profile of FSD201, although we have successfully conducted our phase 1. But here in this trial, we wanted to look more about the AEs, adverse events, and the lab safety assessments. One of the exciting things that we wanted to assess here was the PK profile subset of the FSD201 on day 1 and day 14, which is a single and steady dose in a PK subset of patients. We're planning to enroll approximately 50 patients under this parameter. The exploratory objectives in this case was to evaluate the biomarker levels or the cytokine levels, and I'll explain it a little bit why it is important. So in terms of the study endpoints, our primary endpoint was to look at the patients with disease progression on day 28, and which is defined as a proportion of patients who are either not alive or they are with respiratory failure. So now the respiratory failure, we learned during COVID-19 has pretty much a little bit different definition. And here for this population of COVID-19, we defined as the need for invasive or non-invasive mechanical ventilation, high flow oxygen, or ECMO. Definitions were taken strictly from the FDA guidelines that were released earlier in March of 2020. The secondary endpoints here is to look at the proportion of patients with disease resolution on day 28, which is defined as patients who are successfully made it, they're alive, and they do not require any oxygen or any other therapy, whether they are being treated at home or they're in the hospital. So a little bit about the inclusion criteria of this trial. And that would give you a little bit idea that what kind of target population we were looking at, despite it was not enough that they are presenting with symptoms of COVID-19. So, of course, all of us know we would like to have a signed informed consent form from either the patient or the patient's legally authorized representative. Male or female subjects greater than or equal to 18 years of old. We wanted to enroll the patients who were admitted to the hospital with a positive PCR test or any of the equivalent tests, including the assay test, which is recognized by FDA. Also, the patient has presenting symptoms on day 1, which is either a presence of fever, 100.4 Fahrenheit or 38 degrees centigrade, or they have symptoms like nausea, vomiting, diarrhea, cough, sore throat, or shortness of breath, whether they have it on exertion or at rest, or they are experiencing any respiratory distress. This is actually going to be a little bit changed in terms of our prospective protocol amendment, because loss of taste and smell is also one of the now recognized symptoms of COVID. Another subset that we would like to add and falls under the inclusion criteria is that the patient has presence of moderate to severe clinical signs. And I do get this question a lot: what is moderate to severe? So pretty much, to make it simpler and easier, moderate is when, per FDA guidelines, it's a respiratory rate of greater than or equal to 20 breaths per minute. The oxygen saturation here is very much important, which is greater than 93% on room air or sea level. It might fluctuate for different locations, but this is pretty much that we are going through at this point. Heart rate of greater than 90 beats per minute. Whereas severe, on the contrary, the respiratory rate is greater than or equal to 30 breaths, and the oxygen saturation is less than 93%, which is one of the key markers here. And the heart rate is greater than 125 beats per minute. And the patients have to satisfy either criteria, not all the criteria. Also, in terms of renal functions, they can have normal or mild to moderate renal function after when we conduct the renal function tests. And one of the key things is the patient is able to swallow the tablet, which is a little bit bigger in size, and the tablets cannot be crushed because of the, we want to look at the bioavailability and solubility of the ultramicronized version of the compound. Of course, men whose sexual partners are women of childbearing potential must agree that from first dose of the study medication on day 1 until 30 days, they are actually either having sexual abstinence, there is vasectomy, or their partners are utilizing contraceptive subdermal implants or IUDs or injectable progesterones or any of the following contraceptive vaginal rings or percutaneous patches. All of the above would also be applicable to females, women of childbearing potential. However, for their partners, vasectomy is considered to be as one of the modes of contraception. More so in terms of the exclusion criteria is a little bit stricter here, and that has an impact on the recruitment and enrollment. So in terms of the exclusion criteria, the first is, of course, if it's a discretion of the physician or the clinical investigator that the patient is not likely going to survive for greater than or equal to 48 hours beyond day 1. They're also excluded if they have asymptomatic, mild, or critical symptoms of COVID-19 on day 1. Now, what is asymptomatic? All of us know that they might have a positive COVID test, but they are not representing any symptoms, which I have described in a previous slide. Mild COVID, also, the patients have a positive COVID test. However, they are experiencing symptoms of mild illness, but there are no clinical symptoms that fall under the category of a moderate, severe, or critical COVID-19. The most important here is the delineation between severe and critical COVID-19. Now, the critical COVID-19 is defined where we look at the positive COVID test, but the most important thing is the patient has shock defined by systolic blood pressure of less than 90 millimeter mercury and the diastolic blood pressure of less than 60 millimeter mercury. Either the patient is on vasopressors, and the most important thing here is that they require mechanical ventilation or ECMO, extracorporeal membrane oxygenation. Also, there is humidified oxygen, either it's delivered through reinforced nasal cannula, and it's greater than 20 liters per minute with fraction of delivered oxygen, which is greater than or equal to 0.5. And there are quite a few patients that fall under that category. Also, there are clinical diagnoses of respiratory failure, and that is something that, they are in clinical need for the preceding therapies, or the patient is experiencing multi-organ dysfunction or failure. Not only that, the patient has a documented liver disease except asymptomatic gallstones and screening or day 1. Any active liver disease patients are also excluded. If they have a past history of liver disease, they're excluded. However, the patients with fatty liver disease due to alcohol use are also excluded, but the non-alcoholic fatty liver disease or hepatic steatosis, we are including those patients. The patient has either a Child-Pugh score of greater than or equal to C, which is going to be seeing more in cirrhosis and the underlying chronic liver disease. Patient has immunodeficiency disorder, or they are presenting with Hep A, B, or C, or any kind of viral infection. They have active infection with tuberculosis on day 1, and also they have clinically significant ECG abnormalities at screening or on day 1. And it's also the discretion of the PI that if there is a prior history or it's a current history, whether the patient is eligible to be on the trial or not. They are currently, their renal function tests are not normal. They require dialysis on day 1. Those patients are also excluded. Or if the female patient is trying to get pregnant during the course of the trial. Also, the patient who is planning to donate or bank ova or sperm on day 1 are also excluded. The most important exclusion criteria here is that patients who are receiving alpha blockers or combined alpha beta blockers or antihistamines are also excluded unless they are on a stable dose of greater than or equal to 2 weeks. However, some in our prospective amendment that we have included is that the antihistamines like Benadryls, which are just taken on need basis for sleep, etc., are included. Here, most important criteria here is the patient is also receiving immunoglobulins within 6 months of screening. They're excluded. They have a drug history abuse within 6 months. They're excluded. Also, they're allergic to medications. And if they were part of the previous clinical trial and they have taken the investigational product, basically prior to the first dose in the current trial, FSD 201, which makes it about 30 days and 5 half-lives, because that can affect the biological effect of our investigational product. Or if the patient is going to be transferred in the next 72 hours, and I will go into the treatment duration and schedule in detail. Or the patient cannot read or speak English. However, if there is a medical interpreter or hospital policies allow, if there is an LAR, then we are including those patients. And also, if the investigator, by all means, the principal investigator thinks that it's not in the best interest of the patient to be in the trial, then those patients are also excluded. I do get a lot of questions about the concomitant medication. So all the medications are included, as long as they are for their pre-existing conditions and they don't follow under the prohibited therapies that I just presented in my previous slide. So the treatment duration, it's a pretty straightforward treatment duration. There's a screening period that lasts up to 7 days. There's a treatment period from day 1 to day 14. The follow-up period is on day 28, and the safety follow-up period is 32 days after day 28, which is day 60. And also the 2-day dose interruption is actually allowed in case if the patient progresses to mechanical ventilation or ECMO falls under the critical criteria. And if it's more than 2 days, then the patient will not be eligible to stay on the trial. So study drug dosage and administration, there are, as I said, there are 3 arms. And I did mention it before that the arm 1 has 600 milligrams of tablet we're giving twice daily with a matching placebo tablet. And arm B, we are giving it 2 into 600 milligrams of tablets twice daily for oral administration. And the third one is totally the naive arm, or 2 into placebo tablets matching 600 milligrams of FSD 201. That's also twice daily for oral administration. A little bit, which my interest, and I think most of the people who are watching me today have an interest on this in this compound, is that what is actually ultramicro PEA palmitoyl ethanolamide. You would be very surprised that this was first identified in the early 1950s as an active anti-inflammatory agent in the chicken egg yolk. And also, it has 3 major properties. It's an anti-inflammatory, analgesic, and neuroprotective agent. The formulations with that utilize micro-PEA has a kind of a surrounding or the entourage effect that basically impact the endocannabinoid system in the body. The ultramicro PEA has greater absorption and bioavailability, and it's pretty much very has a favorable safety profile, and it's a very effective drug. If you look at the literature in the past, the first clinical trial was done in a European Union, and there is considerable literature that's present that states the efficacy of the product. More than 22 plus clinical studies were conducted in European Union, and 1 million plus doses were administered. It comes under the brand name of Normast and Pelvalin, which is formulated by the company called Epitech. And there were, to your surprise, not any serious adverse events. And this looks like a complicated slide, but I'll try to make it simpler. It's a mechanism of action. So basically, ultramicronized PEA has 3 proposed mechanisms of action. And the first is the activation of peroxisome proliferator activated receptor system, which basically helps in the lipid and glucose metabolism, in short. And there is another activation of type 1 vanilloid transient receptors, which basically prevents the nuclear translocation, also affecting the lipid and glucose metabolism. The most important and the proposed mechanism here is the downregulation of the mast cells and activation of CB2-like cannabinoid receptors. And this is one of the most important I would say, mechanism of action that FSD believes, and due to the considerable literature that's present in the past, that ultra micro PEA stabilizes the mast cells and downregulates the pro-inflammatory cytokines to downregulate or to have an anti-inflammatory response. And I will go a little bit in detail a little later. So basically, this is the wallet card and the blister card that it comes in. FSD Pharma has developed ultramicronized PEA, and you would be surprised to see that the particle size is only between 0.6 and 10 micrometer. And what is actually micronization? To people who don't know, micronization is a technique which basically is utilized to reduce the particle size, which affects the solubility, bioavailability, and of the compounds that are larger in size. In this case, we have utilized this compound for our initial indication, which was for COVID-19. As you all know, that COVID-19 is characterized by a non-exuberant inflammatory response that leads to a cytokine storm. So our compound actually has a proposed mechanism to downregulate the mast cells and has an anti-inflammatory property to avoid the cytokine storm, which is probably associated and most likely associated with acute lung injury in the hospitalized patients with COVID-19. Now, if you talk about the marketing compound, the products contain PEA have been in the market for long. The naive PEA or pure PEA, they come from within sizes from like 100 micrometers to 2,000, micronized PEAs from 2 to 10, and as I said, ultra micronized is the smallest in range. And it's also now being marketed as food for special medicinal purposes for humans. It is manufactured under the brand name of Normast and Palvalin Epitech, P-Pure by J.P. Russell, and it's also actually utilized as a sole food supplement in Netherlands. And one of the other fascinating uses of ultramicronized PE is also that it is marketed for veterinarian use, which is skin conditions, and Innovet makes it, which is retinal. Also, if you're following the FSD Pharma press releases, we have recently entered into a veterinary license where we'll be utilizing this compound to treat conditions in cats and dogs, which is mostly going to be around the GI purview. So in terms of the physician investigators, what they need to know, as I said, that's has been in the market as a food supplement. It's more than 1,500 patients have received this treatment. Also, there were no serious side effects, even when the dose was up to 2,700 milligrams per day. We conducted phase 1 study, which was pretty, it was in Australia. It was a placebo-controlled, randomized, double-blinded study where 48 healthy subjects had participated, and we looked at the safety, tolerability, and the PK profile of this compound, and there were no serious side effects. If you look at the safety information for the assessment of expectedness of serious adverse reactions, as we said, we looked at the cumulative review of open-label phase 1 study, and then with this compound, there were no serious side effects, and we decided to move to our phase 2A of the trials as we thought that this was data was good enough to go into the next part of the study. This is pretty much, I would like to summarize it, that this is summary of safety data in clinical studies. It would give you a little bit of an idea. As I said, it's a neuroprotective, anti-inflammatory, analgesic compound. And mostly if you look at that in the pain studies, there were no serious side effects. Also, one of the fascinating things is that it's now being used in the dental conditions. And the only thing was that you could see here is that there was one case of drowsiness and there was one case of palpitations, and there were no serious side effects actually reported of this compound. In terms of the worldwide licensing rights, except Italy and Spain. We have the overall license in all conditions and all regulatory categories. We have FSD Pharma holds the strong IP portfolio for ultramicronized PEA, and between 2029 and 34, that's where patent is expiring. What our strategy is to bring the innovative prescription products for in terms of therapeutic intent, and we want to collaborate with the stakeholders. And also, the purpose of this compound is to either use independently or with concomitant use of PEA to address multiple disease conditions. If you look at the potential target indications for phase 2 trials, we are hoping that diabetic neuropathy, fibromyalgia, hematological diseases, pain, one of endometriosis, mastocytosis, and veterinarian uses, it would be very helpful. So our portfolio is prospectively looking pretty good. So in terms of the clinical development programs at FSD, the phase 1, as I said, in the earlier quarter of 2020 was done. And this trial particularly is expected to stop enrollment by September. And we would be closing the study by the end of this year. So in terms of, this was a great compound. We all know about COVID-19, and what were the recruitment challenges or barriers that we actually faced? And I think most of the people who are here now would be in the same boat. So the first thing is the capital and human resource allocation at the research hospitals. I have, as Dr. Lakhan has mentioned, have worked for considerable amount of years in the research hospitals, and I know the infrastructure over there. Sometimes it becomes very difficult from the pharma companies or the CROs to partner there and know exactly their infrastructure or the landscape here. When we were starting this COVID-19 trial, many of the hospitals were either understaffed or they had not figured out a way that how the operations of COVID-19 trials will work. They already had a massive clinical research portfolio. And not only that, the budgetary implications were twice. The other thing that we came across was the activation timelines at the academic facilities. And for example, from the point of PSV to SIV, there's multiple steps that are happening. That's budget, CTA, clinical trial agreement, and institutional review board meetings, which were happening at that time at regular pace, and there were no other guidelines. So we wanted expedited approvals, and it was difficult for us to work within or less than 90 days timeframe, which was very aggressive because these were for the COVID-19 patients, and some of the hospitals were already influxed with a lot of COVID-19 patients at that time. As I said, the critical volumes, we were looking at the demographics and the statistics, and the exact patient population with moderate and severe criteria was a challenge. Not only that, there was an ever-changing standard of care landscape and patient eligibility. Like we have gone from ivermectin to hydroxychloroquine to remdesivir to dexamethasone and to actually emergency use of Beva and Tizo. So it's a lot. And at that time, we were not sure how this disease is going to take the course and what are we expecting. So also, one of the major challenges that we had was the lack of initial guidance by the regulatory authorities, because FDA came up after some time, and we do appreciate that they did a great job of putting things together afterwards. But initially, it was very difficult to see if there are any major deviations, protocol deviations, or minor deviations, or how the things are going to work. Are we going towards televisits, or if the patient gets discharged before 14 days? So these were all the things that, from as a sponsor perspective, we were not sure. Because we felt that the intensive care unit trials are much different than the traditional clinical trials, and they have their own challenges. However, we were also looking at, in terms of the feasibility statistics for COVID-19. Before that, if it was a cancer trial, if it was any of the diabetic neuropathy trial, we knew the statistics were there. But for COVID-19, we did not know where it was a hub or like the hotspot of, the patients that we wanted to accrue or enroll to our trial. So that was the major challenge that we went into. And last but not least, decentralization versus decentralization approach. And patients were we were looking at the trials for all these years as a centric approach versus when COVID-19 and the pandemic crisis happened, we wanted to see that ICU trials or intensive care unit trials are way different. We had issues of how are we going to do the clinical trial monitoring, and what is the whole portfolio going to look like in terms of compliance, and how we are going to get engaged with our stakeholders, especially the physician investigators or the nurse or the coordinators. And especially in this trial, the discharge was before 14 days. So what are we doing? So successful strategies that what we looked at was we actually drafted the protocol per standard of care latest guidelines. We kept it in a way that was physician and the patient friendly and also following the GCP/ICH guidelines. One of the key things that me and my team did was that we collaborated with the key stakeholders. Whenever we were doing the PSVs, we had a considerable discussion on a lot of inclusion-exclusion criteria. That's why we had already a couple of amendments in the protocol. There were a few things that we thought from our perspective was okay, and we did not think about those things. And when we spoke with investigators and the operational team at the hospitals, we actually modified our protocol and our operational strategies accordingly. We looked at the decentralization concept. We wanted to make sure that we are a collaborative partner with the hospitals, because the purpose of this trial is therapeutic intent under Code of Federal Regulations 312.2. If the patient gets discharged before 14, are we giving them, they probably are, for example, 65 years old, they cannot use the e-diary. So how are we collecting the data? Okay, there will be televisits. The nurse coordinators are going to call them and, fill the case report forms or the CRFs and accordingly. So I think those were the little changes that were might sound very minor at this moment, but those were considerable and could have impacted the data. So we took the real time decisions and moved forward with that. Also, the best thing about this protocol, and I think I have, I'm not saying it because it's part of FSD Pharma's protocol, but we did get considerable great feedback because this is minimal number of research mandated procedures in this protocol. And all the activities are revolving around the investigator's standard of care. One of the things was that ultramicronized PE is a safe compound. And we looked at it, although we were moving towards our other clinical conditions, but when we looked at it, we looked at the anti-inflammatory and the cytokine storm that is associated with COVID-19 and how this can be the best study in times of pandemic crisis. Also, the one of the other successful strategy was the schedule of assessments, and the study treatment period only lasts for 14 days, and the compliance of the patients were much better. One of the other things was that we knew the landscape of COVID-19. We were keeping very much adept with the FDA guidelines and what was happening in the world, and especially our clinical investigators. So it was one of the medical affairs team, the great job that they did was to come up with the strategies that how COVID-19 is now evolving and the standard of care is evolving. And we kind of moved towards in that direction. For the feasibility statistics.
I just wanted to interrupt here because we're going a little bit over time, but these are fascinating learnings. Yes. So perhaps if you could sum up in the next minute, that would be great. Yeah.
Yes. Thank you for the heads up. And I think I will just take a minute. So one of the lessons learned here was that we had effective communication with the academic hospitals, and we knew exactly what the ever-changing landscape of standard of care was. We also utilized non-conventional methods of recruitment. I used to personally call every physician and work through them through the process and my phone was like more of a pager, and it is still a pager. Like everybody can call, the research team can call and ask questions. We looked at the risk management. We knew exactly what was the issue. We identified, evaluated, and mitigated the risk. I think this has helped us in, we exactly knew that at that time the data was not available. So we wanted to have the use of real world data versus real world evidence. And I think right now we have successfully recruited a lot of patients, and we know that this trial is going to, when the publications will be there, I think it would have a positive impact. Also, we accurately forecasted the budget, and we knew exactly what was required for this trial in terms of operations and compliance. And I think it is good to go. We are planning to enroll in Latin America and Bulgaria, and I think it's a fascinating compound.
Excellent. Thank you so much on behalf of the industry, right, for supporting in such a volatile, unstable time therapeutics for a condition whose diagnostic criteria, testing, and therapeutics are changing, and all of the amendments and personal touch points that you've done. So, without further ado, we can move on to the next topics. But thank you for sharing your insights.
Thank you. Thank you for having me.
And before we move on, I think the next speaker is setting up shop with his connection. So we can take one question for Dr. Huma from Dr. Vladimir. He said, "Hi, Dr. Huma. Do you use some advanced statistical techniques for analysis patients?"
Yeah, I think that's a great question. And we did utilize some of the statistical techniques, but most of them were actually more of partnering with our partners who actually provided us with the data. So I think it's important to know who are those stakeholders in our pharma and CRO industry who can help us with that. So I think we are still looking for more. I would say that we did not know initially, but we have utilized those techniques, but we are always open to look for better techniques and the statistical analysis, which is a fascinating area.
Perfect. Thank you so much, Dr. Huma. And we will be going to the next session, which is Dr. Abid Singh.